Good morning, Professor Nyonis.
Morning.
Good morning.
Prof.
How are
Sami,
you?
how are you?
Everything is fine. Thank you very much for your efforts coming today.
Thank you. Thank you. You are welcome.
Yeah, just 2 minutes. We are waiting for the other audience to join us, please.
All right. Even more blessing, I think she's also trying to join.
Yes.
I think maybe the next 2 minutes she'll be able to join.
So now we are just on time. Shall we start, Professor Nyoni?
Thank you, Prof. Thank you, colleagues, for joining this meeting. I'm Dr. Stephen Yoni from Chino University of Technology in the chemistry department. And we also have here More Blessing Due. More Blessing Due, she's a PhD student at Chino University of Technology under my supervision. So we are currently working on trying to find solutions in drug discovery through natural products. And Zimbabwe is blessed with quite a number of ethnobotanical plants that we can tap into in order to discover drugs from these natural products. So in this presentation, we are mainly focusing on natural products for anti-diabetes, and these plants, they are also readily available within our country, Zimbabwe, and we thought it wise for us as researchers at Chino University of Technology to tap into that so that we use these natural products for drug discovery and development, not only for anti-diabetic, but for other ailments. So today I have Mrs. More Blessing, who is going to make the presentation of part of the work she's doing at Chino University of Technology. So More Blessing, are you there? More Blessing, can you hear me?
I think she's still she is not online. Yes. Can we send her?
Hello?
Yes, More Blessing.
Hey, Doc.
Yes, good morning.
Morning, Doc. How are you?
I'm fine. Yes, I think our time is up, so we can now start our presentation. May you proceed with your presentation.
Yes. Stop sharing.
May you give me 3 minutes?
Yes.
3 minutes. I'm shifting laptops. The other one has problem. Let me use a flash to to change my slide from my PowerPoint from the other laptop. The other laptop is having problems. Yeah, give me 2 minutes.
Okay.
Give me 2 minutes. I'm transferring my presentation from the other laptop with a flash because the other laptop is giving problems. How are you, Prof. Same?
How are you? Thank you very much for your efforts. Thank you for joining us today and sharing your work with us today.
Yeah,
Would
thank you
like
so much.
we would like actually we started very early, so we'd like to say, to welcome all of the attendees today with us. So until Mrs. More Blessing is ready for the presentation, I'd like to, by the name of NAPRECA, I'd like to thank you very much, Professor Nyoni, for the efforts of the chapter in Zimbabwe. Actually, you are doing a great work, one of the very active chapters in NAPRECA. We witnessed in 2023 the International NAPRECA Zimbabwe at the University of Harare Science and Technology Hub. It was very well organized at that time. So we thank you very much for the efforts of you and your team actually in the chapter of Zimbabwe.
Thank you, Prof.
Which one?
Colleagues, we are very sorry for this technical hiccup.
Okay. The presentation is ready now.
Great.
Can you put it on slideshow?
All right. Yeah, all right. I'm trying to do that. Okay. All right. Are you getting it now? It's now on slideshow.
Yes, you can proceed.
Okay. Good morning, everyone. My name is More Blessing Dove, representing NAPRECA Zimbabwe chapter, and I'm here to present the topic called Unlocking Pharmacological and Therapeutic Properties of Anti-Diabetic Ethnobotanicals. My co-presenters are Dr. Stephen Nyoni and Mr. Sifas Mawere. Can you greet the audience?
Greetings, colleagues. You can proceed.
Okay. I am a lecturer at Joshua Kabuko Polytechnic and also a Chino University of Technology PhD student. Focusing on anti-diabetic medicinal plant. So I am here to present the topic on the pharmacological and therapeutic properties in anti-diabetic plants. As part of introduction, we are saying diabetic mellitus is one of the global causes of high morbidity and mortality rate. It is a disorder associated with insulin and action responses, and we have 4 types of DM, which are very dangerous. Number 1 is type 1, which is called insulin-dependent diabetes. Then type 2 is non-insulin dependent diabetes. Then we have the GDM, gestational diabetes, which occurs on the end-certain of pregnancy and usually disappears after delivery. Then we have the serious and dangerous one again, which is called the secondary diabetes, which develops as a result of endocrine disorder. For example, after hormonal disorders, genetic syndromes, and complications due to medication. Long time taking of medication can cause someone to develop secondary diabetes. Why are we worried about diabetes? Because it is a burden to the country's economy and especially societal and family economy. We are saying long ago, traditionally, diabetes is known with several traditional complications such as retinopathy, peripheral neuropathy, stroke, and coronary heart disease. But with the emergence of this secondary diabetes and type 2 diabetes, we find that there are now many more complications like cancer, functional disability, affective liver diseases, which cause very high morbidity. So what we are saying is there is no cure. According to there's no cure of diabetes, though we have conventional therapies like glucose inhibitors, insulin sensitizers, like the most common drug, which is called metformin. They are there. Insulin injection, they are there, but they are very expensive and they have several side effects. These side effects include drug resistance, development of secondary diabetes, and other complications. There are also another therapies which are called organ transplant, where someone is given a transplant of a organ when it has a problem like liver, kidney, and the pancreas. We can also have a therapy of changing diet and lifestyle, which is very expensive. So for people to overcome this problem, we advocate to have the use of plant-based formulations, which we are saying that's the way forward and the way to go, because these plant-based formulations are very natural, easily available, accessible, safer, and efficient. The main objective of this study is to establish the pharmacological and therapeutical properties of anti-diabetic. Specifically, we'll be identify the ethnomedicinal plants used to treat or to manage diabetes. We'll also evaluate the role of anti-diabetic plants in DM management and to validate the other healing qualities in diabetes. And the methodology which we studied specifically the Matobo district in Matabeleland South Province in Zimbabwe, where we used the mixed methods approach, specifically the exploratory sequential design, where we started with the qualitative designs followed by quantitative designs. We start by surveying 100 participants, especially the traditional medicinal practitioners in the Matabeleland province and the elderly people in the community. We used the interviews, observations, and field survey, where the TMPs were asked the questions on the medicinal plants used in treating diabetes, how they are being prepared, how they are administered, what are the conservation measures, and what are they doing in in treating, is the treatment longer, the duration of treatment, how is it? And the observation was done in the collection of plant samples, and they went observing the traditional healers in treating their patients. And also the plants were collected and were verified at the botanical gardens. We also did a literature review survey where we used the Prisma flowchart to cross-check all these plants which were identified by the traditional healers, and also they were also taxonomized by the Harare Botanical Gardens specialist. We verify them in literature and see, are they also used in other countries or other districts or other parts of the world in treating diabetes? And our answer was yes on that. And we also find out that what are the other therapeutic or pharmacological effects we have in these plants. Then to support our information from the survey, or to validate the information we get from the survey, we did experimental design where we used wet lab experience, in vitro studies, where we do preliminary phytochemical screening using qualitative methods, and also we do quantitative method where we screen and profile the phytochemicals in the plants. We also did for us to have the pharmacological properties and their therapeutic properties, we also did what we call computational chemistry, where we use tools like pharmacokinetics, molecular docking, molecular dynamics, and bioactivity prediction. So one of you might wonder, are there medicinal plants which are used to treat diabetes? If they treat diabetes, can they be used to treat other ailments? Let's just sit back and relax. This presentation is going to tell us from our survey. We managed to collect 97 anti-diabetic medicinal plants from Matopo district. They belong to 77 genera and 49 families. Indigenous plants were over 59, and the cultivated plants were 40. The Fabergé was the dominant family species, as you can see in the diagram. Then we also saw that roots, the main people with the diabetes.
Somebody is blocking your cow.
They used the roots as the main parts in the preparation. As you can see, it has 52%, and it is followed by leaves, which means the main type plants used is roots, and the plants which were so dominant in the area, they are perennials, followed by biennials and annuals. Then the plant growth forms, most of the plants we discovered are trees, followed by shrubs and herbs. Then during the survey, we also did the quantitative analysis using ethnobotanical indices, where we use user value, and we find out that these plants are also used to treat various diseases and also for various purposes in the world, in the rural livelihood. For example, to make timber, firewood as hedge, to make beverages, to make skin protection lotion, to make shampoo, or to decorate, or as floor polish. That this information also drives us to see, so if these medicinal plants they have so many purposes, which means there is something important in them. That's when we went on to verify these using lab results. As you can see, this slide is showing us that our plants were were cited in various literatures several times, especially the Opuntia ficus indica is cited 17 times, and Tamarindas 16 times. After we verified it, we did the phytochemical analysis. We only managed to do sample extraction of 16 plants where we used different extraction method for us to verify the phytochemicals present in the plants. And we find out that the plants which we extracted and did the preliminary test were, for example, by dense polosa and Xylem capense, as you can see in the list. And it shows that the phytochemicals which we are talking about, qualitatively, they are present. We also managed to do the GCMS with the 6 plants, namely Xanthoclum capense, Opuntia ficus indica, Aloe zebrina, Elephantodrisa, Cissus quadrangulus, Diceracrum sinusoides. And after GCMS, we also did quantification of the phytochemicals. But as for now, we managed to only do the flavonoids and the phenolic. Other compounds are yet, other bioactive compounds are yet to be quantified. As you can see, our Xanthoclum capensis has a total flavonoids of 197 and 73 phenolics. Aloe is 161 and 65. By doing this, we discovered that the phytochemical analysis together with the GCMS has managed to give us such groups of bioactive compounds. Why are we worried about these bioactive compounds? It's because if we talk of flavonoids, flavonoids are very useful in managing diabetes because they suppress glucose levels. They help in delaying the progression of anti-inflammatory diseases like cancer. The alkaloids are also important because they are antioxidants and they increase the glucose uptake by the cells. The saponins are very important because they stimulate insulin secretion in the body. Then the phenols, they improve the phenols and the terpenes and the glycosides, they improve insulin sensitivity. So what we can see, these compounds are very useful in the managing of diabetes. And in our plans, in the 6 plans which we did GCMS, quantitative phytochemical analysis and qualitative phytochemical analysis, we found all these groups of phytocompounds. Then we went on to do computational analysis, where at this stage we managed to start Xanthoclum capensis, which is known in Zimbabwe as in vernacular language in Zimbabwe as Mukandanyoka in Shona or Umnyungwane in Ndebele. We find out that in the Z capensis, there are 125 phytoconstituents which were extracted using Xen, and 205 phytoconstituents which were extracted using ethyl acetate. From these compounds, we did what is called pharmacokinesis, where we managed to filter the phytoconstituents so that we left with those which are very useful for our diabetes. So from the list of 125 plus 205 extracted from the extract, we first did what is called filtering basing on CP3A4 inhibition. And CP3A4 is an enzyme found in the liver, which is responsible for metabolizing certain drugs and substances. So we find that some of the phytoconstituents which we found there, they inhibit this enzyme. So if the phytoconstituent can inhibit this enzyme, which means it is dangerous for managing disease. So we discarded all those phytoconstituents which violate with the CP3A4. Then after the CP3A4, we were left with the 24 compounds. After the toxicity test, we also did the toxicity test, and we were left with the 24 compounds, which we saw that they are non-toxicity. By this, we used the Lipinski rule 5, where it is said that the molecular weight of the compounds for it to be a target drug compound should not have a molecular weight of 500, molecular weight of 500, and also LG50 should be 50-50, 5,050 milligram per kg for it to be considered as non-toxic and to be in the level class 6 of toxicity test. After that, we were left with, we also did the BBB permeant, which is the blood-brain barrier, where we tested our 24 compounds again to see if they do not penetrate in the nervous system. What we are saying here, we are saying if the compounds can penetrate the central nervous system, which means that the phytoconstituent is very dangerous because it can cause neurotoxicity. It can damage the brain. So after that, the blood-brain barrier, we were left with 12 compounds. From these 12 compounds, we identified that there was a duplicate. There was one which was the extrin extract and both extrin extract and ethyl acetate extract, the beta amirin. The beta-amirin these are the chemical structures, an example of the chemical structures of the constituents we used for docking, proved to be identified by both as extract. We selected it for further analysis in our journey. So before we did the further analysis, we first did the molecular docking, where the 11 compounds were tested against the existing or conventional 7 drugs used for diabetes. These drugs were metformin, acarbose, netaglinide, pioglitazone, stagliptin, pinagliflozin, and insulin. And we find out that in this molecular docking, we find that all our phytoconstituents, they have the same mechanism with these, their venous score is almost the same as those of the conventional or allopathic drugs, which pharmacies. As we can see, the venous, during our venous score, it is evident that the more negativity, the binding affinity, the stronger the phytoconstituent, and it can be a good potential for formulating drugs. So we use the beta amyrin to see what is being done by beta amyrin as an example to manage diabetes. We compared it using acarbose and pyroglutamine because and one can ask me why we didn't use metformin. We didn't use metformin because it is known that metformin is the main ingredient in metformin is Gallica officinalis from a fresh plant, which we are now saying, of course, metformin has plant ingredient in it. Why can't we test the other ones, which we don't know if their ingredients are from plants? That was how we discarded metformin in our study, in our molecular, in our computational analysis. So as we can see the comparison between the acarbose and the beta amyrin, we can see that the beta amyrin also have the same binding affinity with it has the same binding affinity which is also have the acarbose, and it has many interaction. And as we can see, this unfavorable donor to donor, it has them which also presence in the acarbose. The conventional hydrogen bonds are also present in our compound, which are also available in the acarbose. And with this, we can see that our compound, which we used, is minus 10.9 binding energy, which is very good compared to acarbose, which is 8.3. Acarbose is 8.3, and also total number of interaction, as we can see, our compound is 16, of which acarbose is 12. This can tell us that this is an effective compound, and that can be used as a main ingredient in formulating diabetic drug. We also did the same with plitoglycine, with the beta amyrin. We tested again against that, and we find out that it has the same interaction with the targeted drug, with its plitoglycine. So we find out that our drug has a binding energy of minus 10.9, and this one is minus 8.3, which means yes, I am here. Pioglitazone is minus 8.8, and our beta metarin 225688 compound is minus 10.1, which means our phytoconstituents, or the bioactive compounds found in Z capensis, are very efficient, and they are potential drug ingredients that can be used, because as we can see, the interaction types are of those in our conventional drugs. We also did the molecular dynamic simulation, where we tested our beta amyrin. How does it simulate the actual protein, which is called the pancreatic amylase, which means we tested it against acarbose. As you can see on the graph, the fluctuations of beta amyrin are showed in orange or red, and those of the acarbose, pancreatic amylase are shown in blue, which means our fluctuation, as we can see on a vertical, on y-axis, they are less than 3. So the rule of RMS, it says, of molecular dynamic simulation says, if the fluctuation are less than 3, it means the compound is stable. So with this, we can conclude that the beta amyrin is very stable and can be used as a drug ingredient for treating diabetes. Then we can, from this, the pharmacological properties which we deduced from this is that
the time,
anti-diabetic.
the time, please, Mrs. More Blessing. Just you have 1 minute to finish, please.
We also find out that there are many pharmacological effects which are found in the properties which are found in these medicinal plants. We also did the therapeutic, and we find that the beta amarin, as you can see it here, it also competes with the other drugs. And we can see that the beta amarin is also can be used, is antiviral, is analgesic, is anti-inflammatory, anti-protozoal, is also can be used for cholesterol synthesis inhibitor. Our beta amarin has many therapeutic properties, which some of the drugs which are used in treating diabetes management they don't have. So we can say that these medicinal plants used for treating diabetes are very efficient, and they can be used as drug targets in making drugs which are very cheaper and available. So we recommend that the further research should do the efficacy of the plants, and we are also going to carry forward by formulating the drug using these phytoconfines so that we validate it in vivo and in vitro. So I encourage further researchers to do the same with other plants so that we promote modern drug discovery using indigenous plants. So, but the take-home message from me is plants are life. Let's conserve and preserve them. And these are the references used. And my acknowledgement, I wish to acknowledge my supervisors, Mr. Mawere, my NAPRECA president, Prof. Same, the Matopo residents for giving me to do their survey in their area, the Botanical Gardens, JMN Research and Innovation Committee, and lastly, but not least, my spouse and family. And I thank you. I can leave this time for question and answer session.